Welcome, everyone. Good afternoon. We're so delighted you're here with us today. And I should be saying welcome back to our Probable Meets Possibles online series. Last May, the Bell Museum and the College of Biological Sciences launched a series of conversations to bring together University of Minnesota scientists in bio-based fields to talk about the probable challenges we face and the possible advances that could have a profound impact on our future. These conversations, which you can find in a playlist now on the Bell Museum's YouTube channel, engaged audiences in exciting conversations about the medicinal potential of microbes, the consequences of climate change on water availability and ice cover, and the business outlook for sustainable products and socially conscious investing. My name is Holly Menninger, and I'm the Director of Public Engagement and Science Learning at the Bell Museum. We are proud to be Minnesota State Natural History Museum and a public gateway to research at the University of Minnesota. Our mission at the Bell is to ignite curiosity and wonder, explore our connections between nature and the universe, and create a better future for our evolving world. We're very excited to bring Probable Meets Possible back online this month and next to engage you and an excellent lineup of researchers in conversations that will explore big ideas and new directions in science, that will allow us to engage each other in some future thinking about what lies ahead for our health, for the environment, for the planet, and in today's case, maybe other planets. We are grateful to our partners in the series, the College of Biological Sciences, led by Dean Valerie Forbes and the CBS communications team, including Stephanie Zenos and Claire Wilson, in addition to our own awesome Bell team. Thank you all. Now let me introduce our special guests for today. Dr. Kate Adamala is a biochemist and an assistant professor in the Department of Genetics, Cell Biology, and Development. Dr. Trinity Hamilton is a geomicrobiologist and an assistant professor in the Department of Plant and Microbial Biology. Our topic today, when life gets weird. Seems very relevant for our times, I think. How can our panelists' research on synthetic cells and extremophiles, respectively, provide unique takes on life that may shed important insights into space travel and extraterrestrial life? This afternoon, Kate and Trinity will take us on a tour of the state of their science and share insights about what the future holds for life both here on Earth and on other planets. We'll have brief presentations from each researcher and then open up the floor to conversation. I want to share a few technical housekeeping notes. Please do use the chat box at the sort of bottom of your Zoom panel to send in your questions and comments directly to the panelists. Our team is on the back end is monitoring and curating those questions for our conversation. You may have also noticed that we have turned closed captioning on, again, also the button at the bottom. If you find that distracting or not of interest to you today, you can hide it by clicking on the live transcript button. We are also recording this webinar and we'll post it online as soon as we can on the Bell Museum's YouTube channel. Okay, so now we're gonna start for real. I'm gonna turn it over to Dr. Kate Adamala from the Department of Genetics, Cell Biology, and Development. Kate, the floor is yours.
Thank you so much. All of the Zoom floor I need. So hi everyone. It's a really great pleasure to be here in my room where I spent the last 6 months talking to all of you. I want to give you a little bit of an overview of the field that I'm working in, and then Trinity will give hers, and then we'll just talk about how that science can be used to do some truly weird stuff. So my lab in Department of Genetics, Cell Biology and Development does neither of those things. We're not geneticists, we don't do cell biology, and we don't work in development. What we do, though, is we do biochemistry of life that doesn't really exist. And I'll explain to you in the next few minutes what does that mean. So we're supposed to talk about weird life here. When you look around on Earth, there's this incredible diversity of life. So you might think life is already pretty weird, right? Except it's hard to believe that from the scientific point of view, there is really only one type of life form on Earth. And so that one life form means one type of biology. So the biochemistry of living processes is the same in all of known life. And the blueprint for life, a blueprint for all cells is the same. And looking at the molecular level, all cells of every organism on Earth that ever lived will have the same strategy for storing genetic information, for growing, and for making proteins and metabolism. If we want to explore other possibilities, make lineage agnostic organisms, weird organisms that work on different principles, could live on different planets, we have to start from scratch. So we have to start from something much simpler than a modern natural cell. And that's the whole point of field that I mean, a field of synthetic cell engineering. We want to build simplest living organisms from scratch. And synthetic cells are bringing the principles of engineering into biology. We can make cells with full control of all of its parts, just like engineering a mechanical device. So now just a very quick overview of what those parts are. So the anatomy of most synthetic cells is fundamentally not that different than anatomy of all known living cells. They're lipid containers expressing genes into proteins. And so the cell is surrounded by a lipid membrane, most of the time made from the same kind of lipids as natural cells. And the membrane also has channels, which are holes in the membrane. Those proteins allow transport of nutrients and signals from the outside, and the removal of waste products from the inside of a synthetic cell. And then, since the main point of having a membrane at all is to separate the self from the environment, so the inside of the synthetic cell from the outside, a synthetic cell can also be made in a droplet, so a drop of water surrounded by an oil phase. And millions of droplets can be made at the same time in so-called droplet microfluidic systems, which we also use. And regardless of how the membrane is done, the membrane is a relatively simple problem. The really interesting stuff is what's going on inside the synthetic cell. The genes, the proteins, and the protein expression, all the metabolism of a synthetic cell. And like natural life, synthetic cells express proteins from DNA genomes. Those genomes are usually relatively simple, up to a dozen genes at a time. And to make a complete cell, the genes need to be expressed into proteins. And so that's kind of similar to what is happening in every known life form. We just do it in a slightly different way. In synthetic cells, protein expression is done by in vitro translation systems, which contains ribosomes, tRNA, RNA polymerases, and other enzymes, and all small molecules needed for translation, so needed for making proteins from DNA. And all those components make a complete synthetic cell and can be designed for whatever kind of cell we need to build, including the weirdest possible applications. And one of the most interesting properties of synthetic cells, especially in the context of seeding weird life outside of Earth, is the fact that synthetic cells can be made according to a precise recipe. And if you have a recipe for something, that means you can digitize it. And digital information can be sent across large distances. So it's possible to send information about making a particular type of a synthetic cell to a remote location. And if you can send something across large distances, that also means you can imagine sending that information to a really remote location, like a spaceship or a Mars colony. And so you can make synthetic cells elsewhere, synthetic cells according to the recipe you developed for a very specific purpose. And that with synthetic cells, we can make those simplest possible lifelike models. We can study conditions necessary for the origin of life, both on Earth and elsewhere in the solar system, which we're going to talk about with Trinity later. We also can use synthetic cells to explore how else life can look like. This is the weird life part of today's meeting. We can make synthetic cells to figure out what conditions are necessary for life to evolve and what conditions we should look for when we look for life elsewhere on Mars, on Venus, on Europa, or on Enceladus, or elsewhere in the solar system or outside of the solar system. And synthetic cells can also have many other applications in research and many kind of practical applications relevant to everyday life. So for one, understanding natural cells is necessary for building synthetic cells. And it works the other way around too. If we build an understandable synthetic life cell, we'll learn a ton about natural life. And similarly, making cells from scratch requires a lot of progress in research technologies, and building new bioengineering tools while having synthetic living systems will be kind of tool in and out of itself for studying and for engineering biology. And some other specific practical applications of synthetic cells are making new biomaterials that are not found in nature, building biocomputers that run on artificial genetic circuits, studying the origin of life, astropharmacy, and help with search for life elsewhere in the solar system, Synthetic cells are excellent biofactories. Also, the ability to engineer cells is very valuable for studying diseases, human diseases and other diseases, and for making small amounts of drugs, which could be the foundation of future personalized medicine applications. And for all those space applications I mentioned, we recently demonstrated potential for using synthetic cells for astropharmacy under spaceflight conditions by a sounding rocket experiment launched from NASA Wallops with payloads shared with another UMN professor, Aaron Engelhardt. And building synthetic life cells is a bigger job than one country can support. And making life from scratch is this giant research and engineering challenge that's unifying international research community. And we're organized in the so-called Build a Cell initiative, which is an international science collaboration that's actually based here at the UMN. And I just want to end with the kind of take-home message that the end goal of our work, the end goal of the whole synthetic cell community, is a fully understandable artificial cell. So basically, ability to create living systems on demand with certain properties, and also existence of unified research, international research community. The efforts we do to build cells from scratch are also efforts that help us unify international community, help us work together, help us do biological engineering as one kind of unified community of researchers from all over the world. And if you think about it, in the last few centuries, we went from engineering in steel to engineering electrons, and now biological engineering is finally catching up. We can build synthetic cells, and we're right now moving on to the next step of using biological parts to engineer new types of life for different applications, weird types of life, whatever kind of cells you need for whatever application. And that's the end of my elevator pitch. I want to thank everyone in my lab who contributed to the work that I'm doing. I want to thank Professor Ellen Engelhardt, all the other professors at the U who are working with me, all the members of my lab, all the funding organizations. And I want to leave you at the end with a very relevant New Yorker cartoon. And thank you very much.
Thank you, Kate. In the transition, I'd like to ask you a question before we kick it over to Trinity. And I also want to remind our audience that we would love to hear your questions for, that's going to be sort of the fuel for our conversation today. So I'm strongly encouraging you to place those questions in the chat box so we can make sure that we are talking about things that are relevant and interesting to you. Just quickly, Kate, one of the questions that I have for you, you talked about the recipes for cells and teleporting those. Do synthetic cells have the ability to replicate or reproduce on their own?
They do not right now. That's a bug and a feature. It's a bug because I would love them to be able to self-replicate, and this is one of the holy grails of our community. We want to make them replicate. But the good thing is they don't self-replicate right now. So we can use them as biosensors. We can use them in medical applications where they don't replicate in an uncontrolled way. And we could use them as biomanufactories, for example, on Mars, while preserving the planetary protection because they won't just go on and take over a planet.
Gotcha. Well, thank you for that. I have lots more questions, and I hope our audience does too, that we'll talk about in conversation next. And so now I'd like to turn it over to Dr. Trinity Hamilton from the Department of Plant and Microbial Biology. Trinity, now it's your turn.
Good. Okay. First of all, yeah, thanks for including me in this round of Probable and Possible. And Kate, I just want to say that your slides are awesome, and I was immediately jealous. Anyway, I'm going to talk a little bit more about cells that we observe on Earth. And in my lab, in the Plant Microbial Biology department, we think a lot about weird life. And so this When Life Gets Weird is perfect for us. And what do I mean by weird? I want to show you a couple of slides or a couple of pictures here. I'm going to hide you guys. A couple of pictures of just a subset of the weird types of life that we think about in our lab, which we call ourselves the Fringe Lab. And here I'm showing you some hot spring pictures, an anoxic sinkhole, acid mine drainage, just a bunch of weird types of life that live on Earth. And the reason that we study weird life for a number of reasons, one of them being that Earth hasn't always looked like it looks like today. Plants haven't always been here. We haven't always been here, but microbes have been around for probably close to 4 billion years. And through that history, Earth has gone from different redox conditions, different ocean chemistry, lots of different changes. And so we think about weird life in terms of what life might have looked like throughout this 4 billion year history. And so a couple of examples here, the New Zealand hot springs, these filamentous, wavy type morphologies that we see. These are sunlit environments where we might expect on Earth today these to be driven by photosynthesis. Instead, they're supported by reduced sulfur compounds. This vial that I show you, I call this the frazzled morphology, which is not scientific, but it does seem appropriate. This is a eukaryotic algae that lives at the very limits, the upper temperature limit and lower pH limit of life. So this thing is living on the edge. This anoxic sinkhole, this purpley mat that I'm showing you, this is a cyanobacteria. This is an organism that both makes oxygen and requires oxygen to live, but it's living in an anoxic sinkhole, so an environment devoid of oxygen. This acid mine drainage, these weird, fluffy, biofilmy, brainy type structures that you see, this is an environment that's rich in iron too, which we think mimics early Earth ocean conditions. The snow, this red color that you see on the snow that we've collected from the Beartooth Mountains in Wyoming is actually red because of the snow algae that blooms on the snow surface. And then along the bottom, I'm showing a couple of different phototrophs similar to the eukaryotic algae. These are phototrophs. These are bacterial phototrophs that are living at their upper temperature limit. And so again, they're making really weird structures and living right at the edge of their sort of existence, if you will. And the last one I'll point out is this tomato soup hot spring. We don't understand much about what's going on in this hot spring, which makes it extra interesting. And we call it tomato soup. I think you can imagine why. But this type of system is the red that you see is, you might think of it as more as like an algal bloom or a cyanobacterial bloom that you've seen in a lake. But this is colloidal iron rather than microbial, planktonic microbial biomass. And so we're not exactly sure what's going on in the system, but it is definitely on the weird scale. And so why do we study life here? Because we're interested in interpreting the evolution of life on Earth through different geochemical and redox regimes. But we're also interested in thinking about if we see life or if we find life on another planet, what will it look like? What is the complete gamut of life on Earth? And how can that help us search for life on other planets? So this is just a quick diagram to get you oriented in terms of the chapters of the chapters of history on Earth. And so what I'm showing you here is this geologic timeline. And so we can break up Earth's history into essentially 4 chapters. And those 4 chapters are delineated or broken up by oxygen levels in the atmosphere. So the graph on the top is the only data I'll show. And it's just to show you the percent atmosphere of oxygen in the atmosphere compared to present day levels. And so what I want you to take away from this is during the Hadean and Archean, the very earliest periods of life of Earth's history, there was essentially no free oxygen in the atmosphere or the ocean. The Proterozoic had much lower levels of oxygen than we see today. And then in the Phanerozoic, sort of much more closer to our history, we see oxygen levels rise to sort of close to current levels. And so there are these 4 chapters delineated by oxygen there. So there's been these sort of drastic changes in Earth's surface and atmospheric chemistry. There's also been periods of multiple glaciations where we have ice sheets that have grown and receded around Earth, sometimes even approaching the equator. And the other thing I'll point out is during the Archean and Proterozoic, oceans were largely ferruginous, so rich in reduced iron. So the very nutrient that limits a lot of productivity in the ocean today was in fact abundant or replete in early Earth oceans. And so if we're thinking about weird life, extremophile life, and thinking about what life looks like on early Earth, we need to think about life that lives in the presence of iron and sulfide as well. So in thinking about other planets or potential habitable planets, habitable worlds, I'm just going to highlight Mars and Venus today. I know there are a lot of other moons and planets that we could talk about. Mars, I want to highlight because we spent, we've invested significantly in getting to Mars and looking for life on Mars, including the current rover that will collect samples and cache them for return to Earth, but also Venus, because Venus has made the news recently. And so it's worth sort of revisiting whether or not Venus might have or had life. So similar to chapters in Earth's history, both Mars and Venus have chapters in their history. And within those chapters are periods of potentially warm, wet worlds, similar to what we see on Earth, that would have had oceans. They would have had some amount of atmosphere. They would have rotated and had both diurnal and seasonal cycles. And so the potential for them to have hosted life that is perhaps similar to ours is there. The next question being, well, there are two questions. Is there extant or is there life on Mars today and Venus? And then if there was past life, how do we find it? And so we can use these weird life forms on Earth and synthetic life, as Kate mentioned, to sort of think about what we can look for and how we can detect life. As far as Venus goes, I would argue that the evidence suggests there's probably not life today on the surface. The surface of Mars is over 450 degrees Celsius. It is very hot. There's a lot of lava flows. It's so hot, in fact, that lead sublimes. It goes up into Venus's atmosphere and falls back down as essentially as lead snow. It's a really wild place. The atmosphere, which is where this potential biosignature was potentially detected. The atmosphere is also quite warm. It's rich in CO2 and has clouds of sulfuric acid. So it's a bit different than any sort of type of environment that we have represented on Earth today. Mars, on the other hand, is it's cold, so it's on the other end of the spectrum, but it does around the equator reach around 10 degrees Celsius. And so there is potential for life, especially in subsurface or subglacial lakes where we have liquid water. So that's sort of thinking about the potential for life on other planets. And then I want to come back and just talk a little bit about a specific project in our lab that sort of relates to this idea of Earth history and then thinking about life on other planets. So we're particularly interested in deglaciations, which are common throughout Earth's history. Here I'm showing you the ice extent from the last 14,000 to 12,000 years ago. The last major glaciation on Earth, and then the extent of ice today. And I think what you can see is that during the last ice extent, there was a lot of ice was actually covering a lot of Minnesota. We might have been under around half a mile of ice where we are in Minnesota. And it also gave us these periods of glacial interglacial oscillations gave us this lovely landscape that is rich in lakes in Minnesota. But now and throughout Earth's history, we have experienced periods of deglaciation. And today, in fact, we're in a period of significant and rapid glacial retreat. And both today and in Earth's history, these periods of glacial retreat have been linked to temperature and CO2. CO2 increases in the atmosphere, CO2 being a greenhouse gas. And I'm just going to show you these two pictures, which I hope will convince you that there is a significant decrease in glacial mass. This is Collier Glacier on North Sister in Oregon in 1914 compared to 2011. And so in about a century time, Collier Glacier has lost a lot of ice mass, and this continues around the globe in all glacial and ice sheet systems. So one thing we think about in my lab is the role of snow algae. So these phototrophs that bloom on snow during the summer months. So they need liquid water like all life, and so they only bloom in the summer. You can think of these blooms as sort of similar to, again, a harmful algal or harmful cyanobacterial bloom, where you see these dense green planktonic biomass in the water. So these are red. They make red, which helps protect them from UV in these sort of high alpine ecosystems. And they bloom, when they bloom, they lower the albedo of the snow. So basically, snow has a very high albedo. Most of the sunlight that reaches snow is reflected back away from the Earth system. Anything that is not snow on the snow surface absorbs that energy and accelerates melt or adds heat to the system. And so snow algae do that, and they do that significantly. So snow algae themselves are contributing to glacial decline. And so one thing we've done in my lab is link the abundance of snow algae to increasing CO2 in the atmosphere. So there's some sort of positive feedback mechanism between increasing CO2 in the atmosphere due to fossil fuel burning that also increases snow algae abundance and thus accelerates melt. So coming back to this idea of like probable, possible, and where we sort of fit in, we're particularly interested in the contribution of snow algae to glacial decline, both throughout Earth's history, and so thinking about these Proterozoic and Phanerozoic deglaciation events, but also as we move forward in human history or in human future, and thinking about what role snow algae might be playing with increases in CO2 due to fossil fuel burning. And then the other thing that snow algae do, which I didn't talk about much, is they promote habitability. snow algae add water to a water limited system. And so if we're thinking about Mars has multiple evidence for glaciations during its sort of warm, wet period. And so if we're thinking about even a very simple ecosystem, you could have a snow algae or some sort of organism on a snow surface or an ice surface that is doing the same thing it does on Earth by lowering albedo, generating liquid water, making biomass, and supporting even a very sort of simple ecosystem. So those are sort of two ideas that we're thinking about. And I'll just end there, and I'm excited to hear where the conversation goes next.
Excellent. Thank you, Trinity. Appreciate that. You want to stop sharing? There we go. We're all together on the same screen. Maybe a quick question for you before we then open it up to to big conversation. You mentioned in your presentation briefly about the Perseverance rover that's on its way to Mars right now, and will be collecting samples from Mars that will eventually get back to Earth. I'm curious, what kind of questions, given sort of the focus of your research, what kind of questions would you ask or study using those samples? Like, are you dying to get your hands on those? And what would you look at if you had access to those samples?
So those samples are probably outside so they're going to basically an old lake bed. And so I think there's the potential for the biomarkers that have been preserved, similar to what we see in sort of lake sediments today. And so there's a little bit beyond what we study in my lab. Most of the things we study have to be living. But I think we can help relate to sediments. Lake sediments do something really cool in that they record both water column processes and sort of landscape processes. And so I think one thing we could help with in terms of my lab is thinking about, were there phototrophs in the water column at some time? Are those biomarkers ending up in the lake sediments? So we might be able to help sort of with depositional or environmental characteristics.
Cool. All right. Audience, I'm so excited to see that you have questions that are coming into the chat box. I'm going to encourage you to keep asking those questions. I have some questions, they have some questions, and we'll have some back and forth. But maybe what we should start with is a question that Bill posed, since we're talking about extreme forms of life and synthetic life. Bill asked the question, what is the definition of life? And so I'd be interested to hear, we're talking about it, and I would be interested to hear your take on that.
You go first, Trinity, because I have no idea.
Oh, geez, yeah. I always joke that we in the lab, we come on the scene, so we study phototrophs, right? So we don't have to worry about much until life has quite a few things figured out. So let me see, life should be able to replicate and record its genetic information. It should be able to generate energy, I suppose. What am I forgetting, Kate?
I mean, that's the problem with every definition of life is you can make up a definition and then you will find an exception from it. Because, for example, you're not self-replicating because you're not going to have a kid on your own.
Fair.
And if you haven't had a kid yet, then you haven't replicated, so you're not alive. And that's the problem with every single definition of life people have is there is always an exception. So that's why I don't believe in definitions of life, because there's not a good one out there, really.
Yeah, and I think the people always bring up viruses, and I think we're finding more and more viruses sort of stealing genes from the host cell, and so now that line is getting more and more blurry too.
Exactly. To me, viruses are a biosignature, but they're not life themselves, which means presence of a virus will indicate biosphere and life on a planet, but virus itself doesn't have to be alive. And that's another thing is when you're asking what's the definition of life in a context of looking for life elsewhere other than on Earth, what you really mean is what is a biosignature. So not necessarily what is alive, because, for example, when Trinity gets her hands on a Martian sample, there won't be any living cells in it, I bet you, but there might be traces of life.
Yeah. Can we talk about biosignatures? Because I feel like that was at the end of September, there was this big splashy news that there was that scientists have found, and I'm grossly oversimplifying, found evidence of life on Venus. Maybe let's start by bringing our audience up to speed and talking about what the news was that was announced, the paper that was published. And then I'd love to hear sort of your hot takes on that finding and the interpretation about that latest research about what folks think are happening on Venus. Don't be shy.
So from what I understand in the paper, the scientific paper, at least the peer-reviewed paper, reported detection of phosphine. And on Earth, phosphine can be made through biological means. It can also be made through other means. So I think what the authors were reporting that they found phosphine and making this connection that on Earth, this can be made through biological processes. And then I think what happened is people got really excited about this and made this connection that this is a biosignature, when in fact, it's a lot more ambiguous than that in terms of what we want when a biosignature. We want something that only life can make, especially for something sort of, I've seen people say like extraordinary claims require extraordinary evidence. And so I don't know that the authors were making an extraordinary claim, but if you were trying to, you would need more evidence or a biоsignature that is more definitive for life.
There's also a little bit of a question of whether they even found phosphines, because they used a spectrophotometric technique that you can when you look at the data that they gathered, it can be interpreted as phosphine, but it could also be interpreted as another molecule that's definitely not a biosignature. So they chose to go with phosphines for obvious reasons, but there is a little bit of a discussion in kind of probiotic chemistry and astrochemistry community right now of whether it's even phosphine at all.
I saw a paper that came out, I think yesterday in Astrobiology that I haven't had time to read. That was basically the title. Like, this is not the detection, not phosphine not detected or something like that. Yeah.
Exactly.
That's fancy. Thank you for interpreting that, because I think as you said, like the headlines were very overblown for the moment, for sure. So,
I just want to point out, in defense of the authors that everyone's complaining about, they did not claim they found life on Venus in their paper. It's a little bit of a tale of how to be cautious when you talk to journalists, because their paper did not mention life on Venus. It was all the press circus that kind of followed it, that christened it life on Venus, and that's what everyone's criticizing. The paper itself did not say it's a life on Venus.
Yeah, I think that is a very fair point to make. Okay, so we have and I kind of have a related question, so bear with me as I try to tease this apart. So think about, like, NASA has this Office of Planetary Protection, and that, like, we've all seen pictures online, like as they're preparing the rovers or the spacecraft, that like people are in their Tyvek suits and being in sterile rooms to avoid sending spacecraft like out into the universe with microbes from Earth. And so my first question is, like, is that the only source of sending things out from the Earth? And I'd like to hear your thoughts on that. And then it's a related question. Angelica, one of our audience members, was wondering if it's possible for us to launch synthetic cells out into the solar system and beyond. So like let's talk about that.
I can say Earth has been seeding life all over the creation ever since we started having life. I mean, there's life in the atmosphere, in the upper layers of our atmosphere, which Trinity knows much better than me about it. And we keep shedding atmosphere. So the fact that we crashed a few 100 pounds of hardware on Mars and that hardware was probably contaminated doesn't mean that's the only way we could have contaminated Mars. We've been contaminating Mars for 4 billion years just by shedding natural life from upper levels of atmosphere.
Are they, what lives in the upper atmosphere? Are we talking about like bacteria and microbes? What do we mean by the stuff up there?
I mean, I think there's still some debate about what's living versus what is moving through the atmosphere. But yeah, I think this would be mainly microbial or life that can be transported by wind, essentially, and probably smaller and smaller life forms. You can certainly go collect. There's basically like the Goodyear blimp. You can go out and collect clouds filter them, and you'll find bacteria and archaea and spores or whatever in those clouds. So it's definitely being transported. But I think there's still this sort of debate in the literature about how alive it is there.
Do you think, like, have you heard, and I don't want to put you on the spot, but like, so it seems like then there's tension, right? Between that, like if Earth's atmosphere, if we are shedding things and yet we're also trying to protect things, like I sense a tension there. Maybe there's not. And I'd just be kind of curious on your like, if the reality is that stuff is going out, do we need to try to keep stuff from going out on spacecrafts? Like, is that overkill, or is that or is it seem like a reasonable thing to do?
It seems like a reasonable thing to try. I mean, we can't stop necessarily what's leaving our atmosphere, but I think it seems reasonable to not sort of do it more on purpose. Yeah. I mean, I think we have enough issues on Earth to contend with, that we don't necessarily know all of the impacts of ways in which we're impacting different environments on Earth. So taking that show to outer space is probably not the best idea.
Although once we actually send humans to Mars, there's that boat sailed. There's no way we won't contaminate the planet. Which is why it's so important to have sample return missions right now, while the planet still has some possibility of having its own extant life.
Mm-hmm. So what about the flip side? Like, is there stuff incoming? And is anybody measuring that?
There's a lot of stuff incoming. There's a lot of junk that comes in, and some of it even makes it through the atmosphere. But since we don't actually have any solid evidence of life elsewhere, we don't know what's incoming. There is one meteorite that's been found that has what some people suggest is an evidence of Martian life. It's a Martian meteorite that has structures that some people identify as fossilized bacteria. So that's very controversial debate still. People, I mean, that found is, I think, like 20 years old, but it's still, some people think it's an evidence of life on Mars. So yes, there was incoming life, probably dead life, but incoming life from Mars. And some people say it's just a weird natural structure. So we haven't found little green men landing yet. So we don't know if anything actually survived the transport and if anything's really incoming.
I think too, that's part of the analysis that will happen with Bennu, right? Is the sort of the idea is that it's all really old carbon, but if it's not really old carbon, we should be able to tell.
Yeah, could we bring the audience up to speed? So let me see if I get this right. So yesterday there was a spacecraft named OSIRIS-REx that did a touch and go on an asteroid named Bennu and collected a sample, and that sample at some point in the future will be coming back to Earth. And that Bennu as an asteroid is interesting because it's a lot of carbon, right? Like, did I get
it's
that
all
right?
Carbon. That's the most interesting thing, is it's basically has all the solar system.
Very cool. So tell me a little bit more about Trinity, because you were going to explain too that like what will people be looking at with those samples in particular that's germane to this conversation?
Yeah, so the idea being that it's very carbon rich but old, so it predates almost everything that we know of, and so we can get an idea of solar system formation, perhaps, and other things from it. But then one of the things that I was reading about was that they'll try to look for sort of younger carbon. So if there's been carbon that ended up, or life, they'll go through all the same tests that you would do with like a Martian sample or a meteorite sample or whatever, to think about what exactly that carbon looks like and if there's any biosignatures on it. I don't think they expect to find them, but they'll go through the process.
Very cool. So Patrick asked a question, and I think this is looking out to the future, to like long duration human space travel and potential colonization of other places. So Patrick wanted to know if, wanted to hear a little bit more about if and how synthetic cells can be engineered to make drugs for astronauts when you can't store or ship enough drugs out in advance. So Kate, you kind of alluded to that in your presentation. Could you talk a little bit more about that?
Yeah, that's a very good question. That's actually one of the selling points of our technology for astropharmacy applications, is that when you send people to Mars, you're going to pack up a dozen people in a tin can for a few years, and there's not going to be any material contact with them for that time. You cannot predict what's going to be their needs, both in terms of drugs, nutrients, other small molecules. So you cannot pack up for the whole trip. So the nice thing about synthetic cells is that you can make them to be whatever you need them to be. So you can make them in a relatively simple experiment to produce molecules, including drugs, that you need. So if, let's say, 2 years into a mission, an astronaut comes down with a rare form of cancer that requires a very specific biological drug for chemotherapy, that drug will be able to be made in this on-demand biomanufacturing factory that's a synthetic cell. And the reason it has to be a synthetic cell, not a live cell, is because live cells have their own ideas about what they want to be in life, because that's what they evolved to do. So it's kind of an elaborate process to engineer a live cell that expresses a certain molecule. That's kind of the whole field of bioengineering right now, is making bacteria or yeast or other cells that express a certain molecule. And it takes quite a while and a very well-equipped lab to get a cell line that makes certain things. Because synthetic cells are dead, they don't care. So you can make them to produce whatever you want. And so that's kind of the whole idea, rationale behind astropharmacy, is that we can use those really simple biochemical systems to make not just drugs, but also the other small molecules like nutrients or different materials that might be needed.
Maybe related, some of your work on extremophiles, Trinity, like, is there potential applications as we're thinking about like terraforming landscapes or making, whether it's the surface of the moon or the surface of Mars more habitable? Is there a connection there and a potential application?
Sure. In theory, yeah. I mean, I should say photosynthesis, right, is what got us here. The evolution of oxygenic photosynthesis and the ability of organisms to make oxygen brought humans here, and we require oxygen. And so I think in terms of, if you want to think about something like living on Mars, where the atmosphere is mainly CO2, you don't want to live in pods, maybe you start thinking about trying to grow some type of oxygen producing phototrope. And so when the extremophiles come in because it's cold, and so maybe you start sort of seeding some of these algae or cyanobacteria or something. I'm not sure it's a good idea, but it definitely has been sort of suggested before in thinking about, I suppose too, you could think about other extremophiles to help make the soil or make soil for you to, create places where you could grow crops or food or whatever. But yeah, sure. I think oxygen would probably be the main terraforming idea.
Yeah. Kate, somebody asked, kind of related to that, about how important phototropes are, that can you make a synthetic cell that's phototrophic, that can produce oxygen?
Yes, it haven't been done yet, but we're getting there. We're getting very close to that. There is a lot of work on artificial photosynthesis systems, and that's in a context of this whole synthetic cell chassis. So my bet is within 5 years, we'll be able to see a synthetic cell system that's capable of producing ATP and other biomolecules from light. And the nice thing about it is that terrestrial photosystems are kind of limited to the wavelengths that make it to the surface of Earth and the surface of ocean. If we design it from scratch, we should be able to make a photosynthetic cell that could exist under different conditions. For example, use a broader spectrum on a planet that doesn't have much atmosphere. Here I'm going against what Trinity said. We shouldn't drop the terrestrial crap on other planets, but I think we're going to do it anyway, so we might as well plan for it. So yes, we can make synthetic cells, and hopefully we will.
Related, and I should let our audience know, I'm an entomologist by training, so not a geologist or a biochemist or anything. So Daniel asked if can synthetic cells be made to live in Martian regolith? And I understand regolith is the not dirt, but the material,
Dirt
right?
is fine.
The rocky material on the planet. So,
I mean, dirt on Mars is full of this compound called perchlorate, which is really, really not good for life, for terrestrial life. So if one can make an argument that in order to seed life on Mars, we have to make synthetic cells that do it, because terrestrial cells are not going to live in perchlorate. So we've all watched the movie where a guy grows potatoes in Martian regolith. This is not going to happen because of this one compound, because of the perchlorate concentration in regolith. So I don't know what Trinity's plans are and how she's going to make a polyextremophile that lives on perchlorate, but as far as we know, nothing natural can stand those conditions. So we kind of have to make a cell that withstand that on purpose.
Yeah, interesting. Okay, you brought up the Martians, so I have to ask so at the Bell Museum, we are kind of getting ready and the wheels in motion to develop a new planetarium production that will be about sort of long duration space travel, including the trip to Mars. And so kind of to get into that headspace, I've been reading a lot of nonfiction, like fiction, watching movies, like all of the things. And so like I've been dreaming about Mars lately, like I'm not gonna lie. So I need to know, like, tell us what is sort of your favorite sort of like story or thing that you've seen or read that sort of is imagining that possible future of life on Mars. Just curious. Do you have one? Do you want, or do you try to avoid those kinds of things because it's too close to your research and you try to pick it apart?
You go, Trinity.
I feel like I need to read more.
I mean, mine's gonna be really boring. That's why I wanted Trinity to go first. The Mars trilogy, the Kim Stanley Robinson's books. I grew up reading that. I love those books. So that's for me. That's the future of life on Mars. Is not far from what's described in those books.
Yeah, I think I would agree. I'm not up on like more popular culture books now. I'll have to update my reference list.
Yeah, part of it too, I've been watching, there's a new Netflix show that's called Away that is about the journey to Mars. And there's all these different situations, including a situation, Kate, like you suggest, where I don't want to give spoilers, but somebody gets sick. And how do you manage that when you're on that long duration space trip? So I just thought I'd throw it out there since it's been occupying my headspace. I wasn't sure if you allow it to occupy your non-scientific headspace either. I'm scrolling through the questions. Our audience keeps asking lots of questions. Let's see. So Linda asked a question about saying companies are beginning to make meat food products in the lab similar to what you would eat for food. Is that a synthetic process, like a synthetic biology process, or is that a type of cloning process? She was asking for a little clarity on that.
To me, that's one of the best uses of synthetic biology. And I think, especially, when we go out and establish permanent colonies on the moon or Mars, we're not going to bring cows with us. So if you want a steak, you kind of have to make it from cell culture. So all these fake meats right now, I think, are going to be really, really useful once we can't have livestock anymore, or in places where we can't. And just to kind of answer the specific question, cloning means something very specific. Cloning means moving DNA from one organism to another. And what they do with those fake meats is they don't do cloning. They grow cells of particular type, and those cells can be processed. Basically, meat is cells. So they grow cells that are not grown in a cow, they're grown in a petri dish instead, and they are processed to look like a burger. It's questionable whether they taste like a burger, but they sure look like a burger.
Great. Linda has a question for Trinity. She said that she's heard about slow-growing microbes living in undersea sediments, and she was wondering, what are your thoughts about this research, and if or how it may assist your own work?
I get really excited about this research because most of the stuff that we're studying is growing right now. We need it to double quickly in the lab. We need things to happen on, to some extent, PhD timescales or, human timescales to be able to make significant research milestones. But some of the stuff, I think there's mine water in one of these deep mines in Canada that's has been estimated to be billions of years old. And so we just don't get that most of the time in sort of modern environments. So I think we can learn a ton from thinking about rates of evolution, rates of metabolism from those types of environments, including these deep sea sediments. I think they're very powerful in ways in thinking about time that we are only estimating from, we're using computer programs to sort of help us think about evolution and metabolism over this time that now we have sort of these old modern examples. Yeah.
That's great. Somebody, Christopher asked a question that so if we follow Kate's train of thought and that we seed Mars with artificially designed cells, he was wondering if it's possible to barcode them in a way to tell the difference between those synthetic cells and maybe if there are natural cells on Mars. Is there a way to tag them so that we know?
That's a really good idea, actually. If we do that, maybe the planetary protection people will get off our back. You definitely can. This kind of touches on this concept of biosafety and biosecurity too. If you make something like an artificial cell, you have to barcode it, you have to tag it, and you have to put some kill switches into it because we don't want to release something to the environment, whether it's terrestrial or Mars or any other environment. We don't want to release something that we can't control. So we could put those little barcodes in there too. And people do that right now with genetically engineered natural organisms where they put specific sequences in there. So then you can kind of tag which lab those cells came from. So definitely, if we ever make synthetic cells to release on Mars, I would like to put in a little Goldie Gopher logo in there just to make sure, show where it came from.
Wow, that's great. I have a question about how, I think we can't help but, COVID-19 and the pandemic has kind of pervaded everything about our society. And so I'd be curious about, one, how it has affected your work and your research. I know, Trinity, you showed pictures of places, of potentially field sites where you go, and I'm imagining you're not going to those places right now. And then also kind of thinking about the challenges, and then are there any things coming out of it? I kind of thinking along the line of your work, Kate, about, does synthetic biology have any possible connections to treatments or diagnostics or things related to COVID? So I just like to have a little bit of a discussion about sort of where and how the pandemic has pervaded sort of what you're doing and how you're thinking about your science.
You go, Trinity, you were tagged first.
Let me see. Yeah, so I like to joke that it looks like we do a lot of really cool things and go to a lot of really cool places, but a lot of what we do is on the computer, right? So we're generating a lot of sequence data, a lot of sort of these next generation omics type data sets. And so a lot of the time we spend is crunching through that data. And so we tend to almost get ahead of ourselves by going to the field. And so I think in a number of ways, whether we want to admit it or not, COVID has probably forced us to stop and think about what we have and what we're looking at and spend some deeper time thinking about interpreting that data before we ask the next set of questions. That said, it's certainly hard for grad students who are just starting or even in the middle of their program who had planned field seasons. And we were able to get in the field a little bit. Most of the field work had to happen solely, or I had to be okay sending people to sort of small communities. And so there's just this anxiety and this question of is it really necessary to collect these samples this year, and what sort of risks are we putting undue risks are we putting people in by sending folks to different communities or different areas? And so I think for a lot of us, we took the summer off, and for better or for worse, sat in front of our computers and thought about the next set of questions. And so my hope is that our next set of questions will benefit from this in ways that we don't quite appreciate yet.
Same here. We're experimental lab mostly. We don't do field work. We basically just pipette clear liquids from one tube to another, and that is happening. We don't need field work to do that. We were shut down like the entire university for a couple months, but then we resumed on limited schedule with all the precautions, social distancing, and people are wearing masks. It's difficult because our research is incredibly collaborative. We are very promiscuous collaborators, and we want to go to places, to go to other labs, and we have people from other labs coming to my lab all the time. So that came to a stop, obviously. But we talk to people. It seems like we live on Zoom these days, so we stay in touch with everyone. And in a way, this has been a real boost that the whole situation, the pandemic, and the fact that we still don't have a vaccine, we still don't have a cure, that's been kind of on the forefront of people's minds when they think about what needs to happen with investment in science, what needs to happen in development of biology. We like to think that we're this super smart humanity that can, put people on the moon, and we're so good at everything, and then little virus comes in and we don't know what to do. That means we still have a lot of work to do in developing our science, in developing our scientific enterprise, the way we do science, the way we solve problems. And that's kind of what our synthetic cell community is talking about, because we try to unite and we try to work as one research community across borders. So in the context of COVID-19, it's good to point out that there is this incredible international unifying force, driving force behind all of the experimental sciences right now, that we need to get together. It's not as important as whether I get a first author paper or whether someone else gets this paper. It's important that we get the stuff done. And that's, some people always thought that this is necessary, but now that everyone, including public and politicians, are kind of starting to think about maybe science is actually kind of needed. This is, in a way, obviously, it would be better if we didn't have COVID, but if we have to have one, we might as well kind of learn something from it. And that's what we've been trying to learn too. And obviously that's on top of everyone being really fed up of staying at home and not being able to live life like we're used to.
I think another thing that our lab has specifically thought a lot about, and it could be because of our current personnel, but we've thought a lot about science communication. And thinking about, what are we doing and how are we telling people about it and how can we be better at communication? So I think that's also been sort of one benefit.
Mm-hmm. Well, thank you both for sharing your perspectives. Are there any parting thoughts or maybe, I don't know, like big ideas, things you would like to seed into the brains of our audience to get them thinking, or things they should be on the lookout for in the coming years that might be really exciting or advancements in your field? If not, that's cool too. But I figured I'd leave it with the big question, or the big what gets you jazzed about the future? Maybe that's the better question there in your field.
Go Trinity.
Let me see. I mean, I think one thing that is prevalent in my field is these big, massive data sets. And so we're just sort of, the data's coming in faster than we can learn what the data means. And so I think one thing that sort of, I don't know if it gets me excited or anxious, maybe both, is sort of finding ways to harvest all of the good that's in that data. And yeah, I guess I said it already, but then generate even better questions, moving forward. And then I think this, when these samples come back from Mars, that's going to be exciting. We got to play the long game a little bit, but it's going to be cool.
And I'm really excited about just making weird life, making life that terrestrial biochemistry did not make. So just kind of answering what's possible, what kind of weird artificial cells we can make, and what are they going to do, how are they going to look like.
That's great. Well, audience, I hope from your desk, your kitchen table, or your couch, you are giving our speakers a warm round of applause. We thank you both for being here today.